I'm not gonna.
All right, as we go through this today, what I want to do is kind of draw on the board and kind of simulate what's going on here so you kind of pull it all together today. So I'm going to do this, kind of get to this point here by drawing an example here on the board. So your DNA is double-stranded, right? And so I'm going to draw here a double-stranded piece of DNA. And let's say this is the 5' and this is the 3' end, and this is then the 3' and this is the 5' end. So this is your DNA. Your DNA is in the nucleus. All right. So this is in the nucleus. Let's pretend the rest of the board is the cytoplasm. So you have your DNA in there. All right. So the first step in protein synthesis is to copy the instructions from the DNA onto an mRNA strand. It's called a pre-mRNA strand. So what we have to do is separate the two sides of the DNA. So I'm going to do that here. All right, so this is one side of the DNA. This is the 5, so it's the top side. And then I'm going to separate a little bit here. So here's the 5'. So this is your separated DNA. So this is still your DNA. And so let's say we're going to make an mRNA strand. Do I use both sides of the DNA to make an mRNA strand? No. Why? Because mRNA is only single-stranded, right? So in this case here, let's say mRNA is made from the left to the right from this DNA. If that's the case, if mRNA is made from the left to the right, is this part of the DNA or the bottom strand of the DNA going to be the template strand that you use to make your mRNA? The top or the bottom would be the template strand. You want to be brave and raise your hand and tell me what you think? Nothing?
Would it be top?
Why?
Because no, it would be bottom because you make it from 5 to 3', and if you were to make a new strand, it would be starting at 5 would be where the bottom starts at 3'.
That's correct. The new strand has to be made in the 5 to 3' direction. And it has to be anti-parallel, meaning they have the sides opposite. So if we're moving from left to right, then that means the 5' has to be on the left-hand side and 3' has to be on the right-hand side. All right, so let's say I'm gonna actually put some bases on here for the DNA. So this line here represents the phosphate sugar backbone and the bases stick out. All right, so let's say we have, I'm just gonna write some things here, TAC, GGG, TCA, TA, and ATT. So this is my DNA. So what I'm going to do is build an mRNA by adding the RNA nucleotides that will go to this. All right. So if I am building the DNA by adding the nucleotides, what am I? What enzyme? What adds the nucleotides to build your mRNA strand?
Rna polymerase.
RNA polymerase. So I am RNA polymerase, and so I am going to add, to go with this T, to make your mRNA, I'm going to add a base with, nucleotide with the base A, right? And then what goes with the A? U, right? Because it's RNA, so there's no T's. Then it's G. Then I'm going to have C, C, C. Then A, G, and then with the A, we get a U. With the T, we get an A, so A, and then U, and then U, A. And I'm going to put the phosphate sugar backbone here. So what I've just done is made an mRNA strand. So, so we call this the pre-mRNA strand. So I'm going to go ahead and draw that again here real quickly. So this is my pre-mRNA. It detaches from the DNA. The DNA goes back together, and DNA is done here. All right. And so I have here my A, U, G, C, 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 A, G, U, A, U, U, A. All right. So that's my pre-mRNA strand. Can somebody tell me why am I calling it the pre-mRNA strand? This is what we talked about yesterday. When we went over the answers in that packet was this part here. All right, so this is transcription. We've transcribed or copied the instructions. These are the instructions for the gene to make the protein from the DNA to the mRNA. but it's pre because the mRNA is not finished. What do you have to do, Ashley?
You have to take out the introns.
You have to take out the introns. And so, in looking at this, let's say, remember in one gene, there are introns, and what's the opposite of intron?
Extron.
An exon. Can somebody tell me what introns are?
Are they like unexpressed exons, basically?
Yeah, they're parts of the gene or parts of the mRNA or the DNA that aren't used for coding the protein. So therefore, when you look at this, I'm going to draw this. So this is the whole gene, and I'm going to just put it into E's. And so let's say this is an exon, this is an intron. Let's say the rest of it is exons. All right. So the rest, we'll just make it easy so that there's one intron. All right, just to illustrate here. All right, so introns aren't used to make a protein, and exons are. And so, so the next part of this is called mRNA processing. So we are going to do what to the introns in mRNA processing?
Remove them.
Remove them. All right. So therefore, does anybody remember from the notes what is it that removes them? They call this. So basically, what we're going to do is cut here and here on either side of the intron, remove the intron, and put these exons together. They call them splicing them together. So therefore, the thing that does that has a special name. Starts with an S. Snurps are part of a larger, I know, it's fun to say snurps, I know. It's very exciting. Snurps are part of a larger thing called a spliceosome. Yes, absolutely. So the spliceosome comes over here and cuts the intron out. So to get your final, so we're gonna cut this out. All right. So it's gonna cut here and here. All right. So what you are going to get at the end is your final mRNA, and so your final mRNA is gonna have this codon AUG. It's not going to have CCC because we cut that out. So the next set is going to be AGU, then AAU, then UAA. All right, and that would be the 3'. And so this right here this is your final mRNA without any introns. And that has all the bases that is going to actually code for making your protein.
So the snurps break up the spliceosome?
Yes. Yeah. So it's just part of it, yeah. All right.
So you're kind of right.
Yeah. I didn't mean that you're wrong. I said but it's just part of it, yeah.
Does it matter if the letters are like on top or like under the line?
Oh yeah, you know what? After I started writing it, I switched it. Just pretend like I went like this. Like, you know what I mean? All right, sorry about that. All right, I didn't mean to confuse you. All right. So you have to imagine it 3-dimensionally, okay? So it doesn't matter. And so what else goes on so this part right here is the processing part. What else goes on in the processing part that I haven't illustrated?
Is it the methyl cap and the poly-A tail?
That's right, your cap and your tails. So the methyl cap and the poly-A tail. So I'm going to go ahead and just put those there as red, a different color, to show you the cap and the tail there. What's the purpose of the cap and the tail? For protection. Because where's this mRNA going to go? Into the cytoplasm. How does it get out of the nucleus? Nuclear pore. So I'm going to draw a little hole in the nucleus and put like a little door here. All right. And the mRNA will leave and go into the cytoplasm, which is a very scary place for a little molecule because there's lots of enzymes and things that can attack it and break it down. Hence the cap and the tail. All right. So we want the mRNA strand to reach the ribosome, right? All right. So that's where we want it to go. So this part right here, all right, we talked about yesterday. From here to here is transcription. And then from here to here is RNA processing. All right, so that's going to leave and go to the ribosome. All right, so let's go over the answers to the rest of this RNA processing section of this packet here, now that we've gone over and get the big picture in our head. All right, so on here, so this is showing you that, it's showing you the introns being cut out and the final mRNA leaving the nucleus, which is what I kind of just drew up on the board, and it shows you, and more specifically, instead of just the red lines here, the methyl cap and the poly A tail. And so, so number 12, what has been removed for A is the introns. And B, what's been added is the methyl cap and the poly A tail. How do they get out? For 13 is the nuclear pore. 14, the nucleotides are red, and the starting point is the AUG codon. So the AUG here is how the ribosome knows where to start, so it doesn't include anything before it. All right, so it looks for that start codon, which is AUG, which is why I made mine up on the board in green there, AUG. 15, the M's in mRNA is for messenger. Why? Because it literally acts as a messenger, taking the message, which is the instructions to make a protein, from the DNA in the nucleus to the ribosome out in the cytoplasm. 16, it tells you, this one was a little bit more difficult here. 16, the human genome. Genome is, when they talk about the genome of an organism, that means all the DNA and therefore all the genes for that particular organism. So the human genome has about 25,000 genes. So a gene is a section of the DNA, but yet produces 4 times as many, at least 100,000 different proteins. So the question is how is it that if a gene codes for making a protein and we only have 25,000 genes, it seems logical that we'd make 25,000 different proteins because each gene makes a protein. So how do we get many proteins, many more proteins from one gene? All right, so I'm going to draw this. This one I'm going to draw here is not continuing with this picture here. This one I'm going to draw here is just what happens with the DNA. So here's a piece of DNA, a long piece of DNA. Let's say the gene, there's a gene from right here to right here. All right, so this is a gene. It's a section of the DNA. So this DNA is very long, so they might have another gene over here, so we can call this gene A, gene B, and so on. So there's many genes on one long piece of DNA. So let's just look at one gene, gene B here. This gene right here is comprised of introns and exons. So let's go over here. I'm going to put E for exon, intron, just to give you an idea. All right? So when going through, if we're going to do for this gene, go through this process here in cutting out the introns when we make our mRNA. So when we cut out this intron and this intron, we can get this exon, and this exon. These 3 spliced together. This is your mRNA. That would make one protein. I'm going to call it protein 1. All right? But what happens is, what we found, they used to think one gene, one protein, and they used to actually call introns junk DNA. So they used to think if they don't do it, introns don't do anything, it's junk, it doesn't code for a protein, until they found this little piece, all right, that we make way more proteins than we do genes. And what they found out is that introns are actually very important, and that your cells actually can treat introns as exons sometimes. All right, so therefore, over here, if this one instead treats this as an exon and uses it as a coding, then then it's only going to cut out this intron, and now we have exon 1, now we have a new exon, exon 3, and then we cut out the intron, and then we have exon 4. Notice here, this whole section was cut out from this one, but it's here, and therefore can code and make a whole different protein, so that's protein 2, all right? And then we could treat this as an exon and have protein 3, all right? So therefore, every gene can actually code for multiple proteins depending upon what's treated as an intron and what's treated as an exon, all right? And that was a big deal to discover that, all right? And that's how we get, so that takes care of the mystery about how we have less genes than we do proteins.
You change the intron to an exon, but can you change an exon to an intron or no?
Yeah, no. Yes, that's a good question. Alright, and so therefore, we get multiple proteins. Alright, so that's the answer to what number was that? 16. So then 17 also is a thought-provoking. It says, develop a hypothesis to explain the advantage of a poly-A tail. Oh no, it's not this one. That was thought-provoking. It's another one here. Poly-A tail added to the 3' end. So it prevents the mRNA from being destroyed. And there are these enzymes called exonucleases. It tells you up here that mRNA, once it's in the cytoplasm, is subject to these enzymes that start to remove nucleotides from the 3' end. So this enzyme, once this mRNA comes out into the cytoplasm, you have an enzyme that starts slowly degrading this end. All right, and so therefore, the poly-A tail makes it so that the poly-A's, which means many A's here, they don't code for the protein. So the enzyme that's taking apart the little alligator eating it, when it's taking it and degrading it, since it doesn't code for anything, it protects the mRNA. All right, and so the mRNA can reach the ribosome and actually do its job by taking the message there. So then that goes right with number 18. It says different mRNA molecules can have poly-A tails of different lengths. So you can have short poly-A tails. If the mRNA has a short poly-A tail, it's going to degrade faster. All right, does that make sense? The little enzyme's going to get to the mRNA part that codes for the protein a lot faster. Versus if you have a long poly-A tail, that enzyme, once the mRNA gets to the cytoplasm, starts degrading that tail. It takes longer to get to the important part. All right, and so therefore, it's a way for the cell to actually keep an mRNA around in the cytoplasm a lot longer. So therefore, if it has a really short poly-A tail, it's going to make proteins just for a short period of time. It can go to the ribosome, make a few proteins, and then the mRNA gets degraded. So you may need only a little bit of that protein. The longer the poly-A tail, the longer it stays in the cytoplasm, the more protein you can make from it, and therefore greater quantities.
Does the methyl group go in a certain end in the poly-A?
Yeah, so yes. So this is the tail, poly-A tail. This is the Alright, yes.
So the tail goes on the 3'
Yeah.
Yeah.
Okay. Alright, so it goes out in the cytoplasm, and that tail and cap help protect it. And then 18 tells you to summarize the steps. I just went over that with you, so I'm not going to re-go over it, repeat it, but we did have it on the board there. And then your extension questions. So an exonuclease, even if you didn't know what that was, we could tell it was an enzyme. Why? Because it ends in ACE, right? 21, free nucleotides must be available in a cell's nucleus to produce mRNA strands. Where do the free nucleotides come from? So what you have to imagine is this mRNA, when it leaves the nucleus, eventually it's going to be broken down by those exonucleases. Well, what does mRNA break down into? It breaks down into the nucleotides again. So therefore, when mRNA comes out here, goes to the ribosome, makes the protein, and then mRNA is degraded, you have all these little nucleotides that then go back into the nucleus and then are reused to make a new mRNA. That mRNA gets processed, goes out, makes a protein, mRNA gets degraded into nucleotides, those nucleotides go back, and we get recycle them. All right, so therefore the raw materials to make your mRNA is always recycled. And then lastly, bacteria do not contain a nucleus, but yet transcription occurs. It does the same as the eukaryotic cell, except without a nucleus. So how do we use this to support the theory of evolution? Couple of things. First of all, bacteria's DNA is made out of the same nucleotides as ours. So they have nucleotides with A's, T's, C's, and G's, and so on. So that shows relatedness. And then the process of transcription is the same, where the DNA is separated, it adds the RNA nucleotides, builds your RNA, and so on. So that's the transcription process is the same when the enzymes are the same, and so on. So that shows some relatedness because that's all governed by DNA. Okay? Any questions? Okay, so so let's look at the next step. What comes after RNA processing? What's the next step called? Translation, all right? So translation, so I'm going to go ahead, I'm going to add to this drawing here and first and kind of pull it all together for you, and then I'm going to pass back the translation packets that you turned in yesterday, and if we have time we'll start going over that, but I want to continue this picture here while I have it up here. So what happens? Final mRNA leaves the nucleus, and remember that you have two parts to a ribosome. So you have the small subunit and the large subunit. Remember that? All right, so what comes here is the, oh, actually, I'm going to draw it closer over here so I leave myself some room here. All right, so here's your small subunit of your ribosome. So I'm going to make the ribosome fairly close to the nucleus here. So what comes here is small ribosome, and then the mRNA comes and binds to that, and then the large subunit comes. All right, so that's the process. I'm going to draw it where it's all done already because it's easier to do that here. So this is the large subunit. And before I draw my mRNA on there, the large subunit has three kind of spots on it. You guys remember what they're called? Just the letters. There's three. They were drawn like this. One of them is A. Here's A. P. And E. EPA. All right? So, and this is your large subunit. This whole thing is your ribosome. So now let me draw in the mRNA. The mRNA, remember, actually, before the large subunit comes in, it comes in, the mRNA binds to the small subunit. So this is your mRNA. Final mRNA. So this is your 5' end, and your cap's hanging off here. And this is your 3' end, and your tail's hanging off here. All right, so that's what we have. All right. Actually, I'm going to make this a little, well, it's okay. All right, so now, what we have to do is draw in the codons. So AUG is our first codon. The start codon always starts in the P site. All right. So this right here is not coding for anything. So AUG, first codon in the P site, and then AGU is in the next one. Then we have AAU. That's hanging off. It's not in any site yet. And then we have UAA. All right? So this is what we have in our mRNA. So AUG, this is the start codon. The codons in the mRNA is what you use in that chart to determine what amino acid is going to be the first amino acid in our polypeptide chain, because that's the purpose. This is the message to build it. So AUG you would use in the chart. And so for this, we're going to do like where you're going to need the chart. So you might want to get the chart out so that from your notes so that you can look at it, because I'm going to be asking you what the next amino acid is and so on and so forth. So unless you've got it memorized. And even I don't have it memorized.
You guys don't have it memorized yet?
That would be,
I'm like halfway there.
you're halfway to having it memorized? I thought I heard you say you're halfway there.
Wait, what was it?
I'll make it my goal this weekend. 4 days.
All right. So AUG. AUG stands for what amino acid? This is called the start codon, but it actually codes for an amino acid. And we can just use the first three letters. It's MET, all right, which is methionine, which means there's a methionine amino acid floating around out here in the cytoplasm. So the amino acids, remember that we get them in our cells from our diet. So when you eat bacon or something from a pig, you're eating pig DNA, all right? And then you break that DNA down into nucleotides, that goes into your blood to your cells, and these nucleotides are now floating around
And then you get
in your
IVs.
cytoplasm. And methionine is one of those amino acids. All right. Now, we need our first amino acid to be methionine. How do I get this amino acid here to be the first amino acid? How do I get this here to be my first amino acid? It does look like a candlestick, all right? In the computer generated, it kind of looks like this. What is this?
tRNA.
Yeah, this is tRNA, and there are many different kinds of tRNAs. They are a strand of RNA, just like mRNA, but they fold on itself and make a structure that looks like this, where the wick of the candle is where the amino acid attaches, and then this point here, they have what's called an anticodon. They call that an anticodon because it's opposite of a codon. So the tRNA that has the anticodon that goes with AUG would be what? What would be the anticodon? UAC. Now this tRNA is the only tRNA that can bind to this particular codon. And this particular tRNA, the only amino acid that it can pick up is methionine. And there are enzymes, you don't need to know the name of them, they're amino acyl tRNA synthetase, all right? But you don't need to know that, all right? There's an enzyme that puts methionine onto the tRNA. And this, as I said, this tRNA can only pick up methionine. So while there's 20 different amino acids, while there are a whole bunch of other amino acids floating around in the cell, that tRNA only picks up methionine. That's important because this tRNA is the only tRNA that can hydrogen bond with this codon. So this tRNA is going to come over here. So I have my UAC, and then the tRNA comes in here and brings with it the first amino acid, which is methionine. So now I have amino acid 1. I'm starting on the way to building my polypeptide. So then, my second amino acid, figure out what the second amino acid is going to be based upon what I drew on the board. What's the second amino acid going to be in my polypeptide chain? So, how do I do that? I look at the next codon. And you always use the mRNA codon. AGU. You go over here to your chart. AGU is SER, which is serine. All right, so SER. All right, so I'm going to go ahead and draw this one here as SER. How is SER going to get to be the second amino acid and be brought over here? tRNA is going to pick it up, and what's true about this tRNA? It's different than this one. It has UCA. So it has the anticodon that goes with this. So therefore, this is the only one that combines with this, and this tRNA with this anticodon can only pick up serine, so that makes it, without a doubt, the next amino acid is going to be serine. So this tRNA comes over here. All right, so we have UCA. All right, and the next amino acid is SER. All right, so now we have amino acid 1 and amino acid 2. Now, on this, what happens next? Before it shifts. What has to happen to the met and the sur? We're trying to make a polypeptide chain, so the amino acids should be hooked together, right? So how do we hook them together? We break this bond here, so methionine is no longer attached to that tRNA. That tRNA has done its job. Its job was to bring the first amino acid methionine. And then what happens is the methionine connects to the serine. So this bond is broken, this bond is formed. So now I have the first 2 amino acids in my chain, okay? So then, now what? I have 2 amino acids. What happens next? It shifts over. What shifts over? It's as if, pretend that I took this mRNA and pull it over one whole codon. So this codon goes in this site, this codon goes in this site, and now this codon is going to go in this site. So to make it so it's not messy, that we can follow along here, I'm going to draw here another picture of what that looks like. Okay? So here we have, right here, all right? So it's the same ribosome, but now it's going to look like here we have our EPA site. Now when I draw my mRNA, AUG is going to be over here. All right, so I'm going to do AUG here. And then AGU is going to be here. And now my next codon is going to be in the A site, so that my next codon is AAU. All right, so now I have my mRNA there, my 5' and my 3' here, and to make it consistent, and my cap and my tail. All right, so so let's draw in the tRNAs. Now, do you see when this AUG shifts over? It's in the E site. E stands for exit. So what has happened is, do you see that this tRNA, since it brought the first amino acid, and now that amino acid is attached to the second one, this tRNA's done its job, it's done, all right? So why it's called exit is because when this shifts over into the E site, this tRNA leaves. So I'm going to go ahead and draw that tRNA leaving here, all right? Notice there's nothing attached to it, there's no amino acid attached to it, and it has the UAC. All right, so that has left the E site, it's done its job. What is that tRNA going to do now? It's going to go find another methionine, and it can bring a methionine to another, polypeptide chain. Yes.
What happened to the UAA? That was like on the outside, like.
Oh, I forgot to draw.
Okay.
So it's still hanging out here. Sorry, thank you. All right, so it's still hanging out here, it's just not in a site yet. So now let's look at the AGU that's now in this site. So this tRNA shifts over here. So we have the UCA with the tRNA that has the serine and the methionine. All right, so now you have 2 amino acids there. So we're 2 amino acids in. So now, how do I figure out what the 3rd amino acid is? See if you can figure that out. Figure that out, and if you're drawing, figure out what the tRNA looks like, all right, with the anticodon on it, that's going to pick up that.
Does the last step always go from like the 5' and 3' and holes? Okay.
Wait, what one are we doing? Are you doing
AAU. That's
We're doing the 3rd amino acid.
It would just be.
Oh, I was thinking of the opposite of it. Don't you?
No, you take the mRNA.
Okay, take that one.
Wait, so we're looking for the mRNA.
So it'd be
RNA.
Yeah.
You do the AAU. So we look at this next codon, AAU. You go to your chart, and AAU is ASN, right? Is that what you got? And so what we have here is there's an amino acid called ASN floating around in the cytoplasm from our diet. The tRNA that's going to pick up ASN is going to have an anticodon of UUA, right? And this is going to come over here, land here. And hydrogen bond, and it's the only one that can do that. And ASN. What happens now? Be able to tell me what happens based on this bond breaks, this bond forms. And now I have 3 amino acids in my chain. And this will continue on. Some polypeptides, I made it really short, all right? Some polypeptides have hundreds of amino acids, so it will continue to do this, all right? And so then what happens next? I have 3 in a chain.
I just have a question.
Oh, okay.
What bond holds the amino acids together? Is it like a hydrogen bond?
It's a peptide
Oh,
bond.
yeah.
It's a covalent bond. It's not a hydrogen bond. They're sharing one bond. Okay, so what happens next? I pull it and shift over one codon. Okay, so I'm gonna draw that down here. All right, so large, small, 3 sites here. All right, so what is that going to look like? Now look at here, AUG is going to hang off now. So if I draw my mRNA here, this was the 5' end, AUG is now done its job, and so it's not in any site here. The AGU is now in this site. The AU is in this site, and now UAA is in this site. Okay? So this is the 3', and then we still have the cap and the tail, all right? And so that's where how the mRNA has shifted. So let's look at the tRNA. So AGU, this has shifted into this. So notice that this tRNA no longer has anything bonded to it, so it's done its job. It's now in the E site, so it's going to exit, all right? So therefore we have tRNA with UCA shifted and exited. All right, now it can go to find another amino acid, all right, and be reused. So tRNAs are always being reused. The AAU, this one is now in this site here, so we have the UUA tRNA, and now what's attached to it is 3 amino acids. So we have ASN, SER, and MET. So now we have the 3. Now, let's look at the next codon. What's the next codon? UAA. What does that stand for? Yeah, it's one of the 3 stop codons. So if you look at UAA, it says stop next to it. Notice that there's no amino acid next to it. So this does not code for an amino acid. This just means, hey, we're done. We've done building our polypeptide. This is it. All right, and so therefore, we're not going to add another amino acid to this chain. But what does, so there's no tRNA that's going to come here because there's no, it doesn't stand for an amino acid. And so what does come here? Grace?
Water.
Water, and there's something, it's a general name of something that's going to help to kind of disassemble everything. It's a generic name, there's not a specific name. Things are going to be released because they're all done. It's called a release factor. All right. So there's
So
generic
Steve would have
things.
been a
So
better
this
name.
is a release factor. And it does not code, it does not bring an amino acid. What that's going to do is trigger a couple of things to happen. It's going to trigger this bond to be broken, and it's going to release, that's the name, release the amino acid chain. So once this binds here, it all kind of breaks apart. The whole thing goes back to how it was before. So I guess I'm going to go around in a circle here, because I'll show you this. So everything is released, so what happens next is when that release factor binds, the large and the small, large subunit separates from the small subunit. Your mRNA and amino acid chain separate. So now we have our mRNA chain, 5 and 3 primes. This is your mRNA. I'm not going to draw all the letters here. And then we have our polypeptide. So I'll put those in here. MET, and ASM. All right? And so it kind of disassembles. That's why this is called a release factor. It triggers that to all come apart. And so it all disassembles. Now this is your polypeptide, and it can do its job. Maybe it is an enzyme in the cell, something like that. So it can do its job, and the mRNA will eventually be degraded by those exonucleases, and let me put this down here because some people can't see. It'll be degraded by those exonucleases into the nucleotides, which will go back in and be reused to make more mRNA, and so on and so forth. All right, and so that is translation in a nutshell. Was that helpful to kind of pull it all together? All right, and so on. So realistically, you're not going to have just three amino acids to make up your polypeptide. And just to pull in from a previous chapter, that remember that these polypeptides at some proteins are made out of more than one polypeptide chain. Remember, hemoglobin was made out of 4 different polypeptide chains. So they would have to go through this 4 times to get the 4 polypeptide chains to come together, all right, to make a protein. So this really, more specifically, this really should be called polypeptide synthesis because this chain is not always a finished protein. Some proteins need more than one chain to come together, all right? So it may or may not be, all right? Alrighty, so I am going to pass back your translation.
I drew a purple owl.
Instead of drawing all that, you're just like, yeah, I'll sell purple owl. I took like 15 pictures of it, like as she was going through, because I was scared that I wasn't going to get one later, so I just took like a ton.
Gonna take.
Oh, we're not.